So if you don't already have a calculator with you and like a pen or a pencil and some paper that you can write some things down, you definitely want to get those things because Hardy Weinberg definitely requires the calculator, and I want you to be able to be practicing alongside of us to make sure that you're getting the right answers. So go grab that now if you don't have it already. All right. Here they are, the Hardy Weinberg equations and definitions. Okay, so there are two equations that can be used for Hardy Weinberg equilibrium. One, the first one is nice and simple. It's P plus Q equals 1, and I'm going to define all those variables for you in a second. The second one you'll notice is just a perfect square of the first one. Okay, that's where we get P squared plus 2PQ plus Q squared equals 1. Okay, what does P mean? What does Q mean? P squared, blah, blah, blah. What does it all mean? Well, that's defined for you below. And on the AP exam, you would get both equations as well as the definition of P and Q on your equation sheet. Okay, so these are not things that you need to memorize. You just need to know how to use them. Okay, all right. So P and Q, focusing on that first equation. So that first equation is used to look at allele frequencies and how they're changing. Okay, how they're changing in the gene pool. So you're gonna have to bear with me here. I'm gonna explain this a couple times because students always mess it up, okay? But P and Q are going to be equal to the frequency of the dominant and recessive alleles in a population. Okay, so P, I think of P as in powerful. P is the frequency of the dominant allele, okay? On the flip side, I think of Q as in quiet, and Q is the frequency of the recessive allele, okay? So remember, an allele is just one half of a whole. It is one part of an entire individual. You have two alleles for each trait that you have. Okay, so I'm going to grab my poker chips. So if you're homozygous dominant, you have two dominant alleles. If you're heterozygous, you have a dominant and a recessive allele. And if you're homozygous recessive, you would have two recessive alleles, okay? But just a single allele is what we're going to be counting in the allele frequencies for P's and Q's, okay? I'm going to show you an example picture in a second, but when you're thinking about P's and Q's, you just want to be thinking about individual alleles or individual versions of genes and not whole organisms, okay? That's going to become really, really important when we take a look at some of these practice problems and you're trying to decide what you're given and what you're looking for. Okay, more on that in a second when I give you a sample picture. Okay, other things that are defined below are helpful and they're not given to you on the AP exam, so it is something that you need to remember. Okay, so these next three things define the second equation and what that equals. Okay, so we have P squared, that's two powerful P dominant alleles. Okay, and that's going to be equal to the frequency of the homozygous dominant genotype. Okay, so P squared is our frequency of all of the homozygous dominant individuals in our population. 2PQ is the frequency of the heterozygous genotype. They have one P and one Q, right? That's why we call them heterozygotes. So those would be all the individuals in the population that have one dominant and one recessive allele. Okay. Both homozygous dominant and heterozygous P squared and 2PQ are going to look dominant. They're going to have the dominant phenotype. Okay, so for my example in the beginning of this podcast, okay, that would be the red-eyed flies. So if I tell you that you have a certain number of red-eyed flies, that's going to be a combination of homozygous dominant and heterozygous individuals. Okay. Lastly, Q squared, two recessive alleles, is going to be equal to the frequency of the homozygous recessive genotype, or how many organisms we have that are homozygous recessive. Okay. The really great thing about Q squared, and the reason it's going to become your favorite of all the variables, is because if something is homozygous recessive, it's the only time that that thing will look recessive. So from my example in the beginning, if I told you the number of flies that have white eyes is the recessive phenotype, okay? So that frequency would be the same as the frequency of Q squared, okay? Which is something that makes the recessive phenotype different from the dominant phenotype, okay? This is going to make a lot more sense once we start practicing, I promise, and when I show you this next picture. Okay, but something I want you to notice about these equations is that they always equal 1. Okay, so that means our frequencies are decimals. They're parts of a whole. Okay, so if I was trying to take a look at a population and I was trying to count up the number of dominant and recessive alleles in that population, let's say we have one homozygous dominant individual and one heterozygous individual in our population. Okay, if I just want to look at allele frequencies, I would have 3 dominant alleles out of 4 and 1 recessive allele out of 4. Okay, so P would be 3 out of 4 or 0.75, and Q would be 1 out of 4 or 0.25. Okay, and if you look at equation 1, 0.75 plus 0.25 is equal to 1, right? Basically, it's a fancy way of saying if 75% of my alleles are dominant, then the other 25% are recessive, right? It's just making sure that all of our alleles or all of our genotypes are accounted for, okay? Equation 1 is really the only equation that you'll use. Equation 2 is more just there just to define the frequency of the homozygous dominant, heterozygous, and homozygous recessive genotypes, okay? And you'll notice that we have some males and some females that look different and have different genotypes below them, okay? So the first thing that you should notice is that all of the Black males and females are what we would call heterozygous. They have 1 dominant allele and 1 recessive allele, okay? If we were just looking at the sheep, we would just see, hey, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 black sheep, okay? But because we also have their genotypes, we can say that there are 12 heterozygotes in this population, okay? On the flip side, we have 12 homozygous recessive white sheep, okay? Even if I didn't see the 2 little b's below them, if I knew that white was the recessive phenotype, I would know that all of those sheep are homozygous recessive because that's the only way that they would look white, okay? So that's something important to note from the beginning. So if I were to look at this population, in total, I have 1, 2, 3, 4, what did I just say? 24 sheep. So I'm going to flip to a screen really quick. You can, remember you have this PowerPoint that you can pull up on another screen if you want to follow along with that. I'm going to pull up a whiteboard screen very quickly here. So to show you, so we have 24 sheep in that example. Sorry, you have to bear with me. Okay, we have 24 sheep in that population. We have 12 black sheep, 12 white sheep. So the phenotypes, we're 50-50, right? We have 50% of our sheep look black, 50% of our sheep look white, so 50% dominant, 50% recessive phenotype, okay? Now with this, I can take this a step further because it told me the genotypes as well. Okay, so I have 12 sheep that have 2 little b's. Oops, I almost tried to make an A because that's what I usually write. 2 little b's, okay? And my black sheep, I have 12 that have 1 big B and 1 little b, okay? So if I was trying to set these equal to variables from Hardy Weinberg, okay, what I would do is I would tally up how many alleles we see. So you could just count. If you're on your screen and you're looking at the sheep, you can just count how many big B's and how many little b's you see, and we can sort it that way, okay? But if I want to see how many dominant big B alleles I have, that would be equal to P, the frequency of the dominant allele, and how many lowercase little b's I have. We can count them, or we can do some simple math here. Okay, so our 12 black sheep, they're going to have 12 big B's, right? Because they each have 1 big B, and they're going to have 12 little b's, okay? Our 12 white sheep are going to have 2 times 12, or 24 little b's. Because remember, they have 2 versions of each, but theirs just all happen to be little b's, okay? So my 12 big B's, I can write up here. And my 24 plus 12 little b's will give us 36 little b's, make up the entirety of our population. So if I wanted to change this into a frequency, right, which is what P and little b would be Q are equal to, okay, I need to make this the 12 big B's that we have, over the total amount of alleles that we have for our population. So if we have 24 sheep and they each have 2 alleles, then we have 48 alleles total. So I would put 12 over 48, and I would get that a quarter, or 0.25 of my alleles are dominant, and that's equal to P. Okay? If I put 36 over 48, then I would get 0.75, or 75% of our alleles are recessive, which would be equal to Q. Okay, so the frequency of the dominant allele P is equal to 0.25. The frequency of the recessive allele Q is equal to 0.75. Okay, I can also look for the frequencies of the heterozygotes and the homozygous recessives in the population. So 2PQ is what we use to represent heterozygotes. You just have to remember that, okay? If you look back on the slide before, you'll see that, okay? And that's going to be equal to, we had 12 of them, right? 12 heterozygotes, all of our black sheep out of 24 total sheep. So 0.5 would be equal to 2PQ as a frequency. And then Q squared would be equal to 12 out of 24 as well. So we had 12 homozygous recessive sheep, or 0.5. Okay, so that's how we could define all of those variables with a given population, knowing what they look like and what their genes say. Now, usually we don't know what they look like or what their genes say. Here's our problem. So in a certain flock of sheep, stayed with sheep again, okay? In a certain flock of sheep, 4% of the population has black wool, and 96% of the sheep has, that should say, white wool. Okay, so 4% of the population has black wool, 96% of the population has white wool, and we are going to assume that the population is in Hardy Weinberg equilibrium. Okay, if black wool is a recessive trait, okay, ding, 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 so the black wool population is recessive here. What percent of the population is heterozygous for this trait? Okay, that's a lot of words. Your first step always for a Hardy Weinberg problem is to figure out what is given, right, and what you're looking for. So I call that the given and the unknown, okay? What the heck are the numbers that they gave you, and what the heck are you looking for in the problem? If you can figure those two things out, and you know how to square and square root, then you know how to do Hardy Weinberg, okay? So let's figure out our given and our unknown in this problem. All right, so given. So this is where we're going to look at the numbers. So if you reflect back on that problem, okay, so what we need to know is then what that actually means for us. So 4% of the population has black wool, which is recessive, and 96% of the population has white wool, which is dominant. We're going to see this in another problem, but for the sake of an easier start, just know that we don't want to deal with that dominant population because we don't know if they're homozygous dominant or heterozygous. Remember that these organisms, homozygous dominant, and these organisms, heterozygous, they all just look the same. We don't know what their genes say unless we're going out to all these sheep and drawing their blood, which we are not, okay? So that 96% of sheep that had white wool could be either of these things. It's some sort of combo of homozygous dominant and heterozygous individuals. So we don't know much about them. So we are going to ignore them. The thing that we do know, though, is that the 4% of the population that is black, they gotta be homozygous recessive. Otherwise, they're gonna look white. The only reason that you have that black wool is if you are homozygous recessive, okay? Remember, homozygous recessive, if you flip back a few slides, is equal to Q squared, okay? That is just always true, no matter what. So knowing that 4% of our population looks recessive, that means 4% of our population is homozygous recessive, And so we can set that 4% equal to Q squared, right? So 4%, changing that into a frequency, I make that 0.04% of the population is homozygous recessive. That's equal to Q squared. That's what's given to us in the problem. That's what's usable in the problem, right? The 96%, ignore it. What are we looking for here? So if you read this question over again, if black wool is a recessive trait, what percentage of the population is heterozygous for this trait? If you look at the things that I just listed in the chat for you guys, okay, you'll notice that heterozygous, what we're looking for is 2PQ, okay? So the value of 2PQ is our goal of what we're going to find, and we have Q squared being equal to 0.04, okay? Finding the given unknown and unknown first is essential to understanding these problems, because now we know what numbers we have that are usable, and we know what numbers we need to find or what variables we need to find, and then the rest is pretty simple, okay? So next, I'm using a method called the guess method. So we found the G, we found the U. The next step is to look back at your equations, which I just posted in your chat, okay? But equations, or E in the guess method, the equation that you're always going to use is P plus Q equals 1, okay? Because if you can use P plus Q equals 1, you can find P, you can find Q, and therefore you can find any of the genotypes that you might need to find, okay? All right, so we're given Q squared, but I need to use this equation P plus Q. We got to square root it. So this needs to be something that you become really familiar with. Yes, thank you, Tania. So we need to square root this. That's how you get rid of a square, right? It's just like the inverse function of a square, okay? So if we have Q squared and we need to find Q so that we can plug it into our equation, then you need to grab your calculators and square root 0.04, okay? And you can write this out. It's hard for me to write on the whiteboard and switch back and forth into PowerPoint. I'll try to do that for some of our next problems coming up, okay? But for all intents and purposes, you can just grab your calculator, you can square root 0.04, okay? And you should get that Q is equal to 0.2, okay? Q squared is equal to 0.04, therefore Q is equal to 0.2. We have this equation, P plus Q equals 1, okay? So now we can find P, right? If P plus Q equals 1, P plus 0.2 is equal to 1 then. And if you think about it logically, because some people just like to actually think about what they're doing, okay? If 20% or 0.2, right, 20% of your alleles are recessive, then the other 80% must be dominant. Otherwise, we have aliens in our presence or something crazy is happening, and that's just not happening. So 20% is Q, then 80% better be P. And if you do the simple math there in P plus Q equals 1, you will find that to be true, okay? So P is equal to 0.8. Q is equal to 0.2. If your P's and your Q's don't add up to 1, you did something wrong. So back up, see where you messed up, and try again, okay? All right, so last step here is to solve. So remember, you may have forgotten when we're on this like winding road to nowhere right now, what we're actually looking for. And what we're actually looking for, unknown if you go up to U, is the percentage of the population that's heterozygous, okay? Remember from looking at the chat, heterozygotes, the frequency of them is equal to 2PQ. And now we have P and we have Q. So this just becomes the simplest math problem ever, okay? So we can plug in for P and Q, okay? And we will get 2 times 0.8 times 0.2, which is equal to 0.32, okay, or 32% of the population being heterozygous, okay? So to back up or rewind what we did there, we found what we were given. We were given that 4% of the population was homozygous recessive, or looked recessive, which you know as brilliant biology students means that they're homozygous recessive. We used that to find Q squared, okay? We knew that we were looking for 2PQ because the question is asking us the percentage of the population that's heterozygous. We know that we're going to use equation 1, P plus Q equals 1. So to make that usable for ourselves, we found Q from Q squared by square rooting. We plugged into P plus Q equals 1 to find P, and then we plugged into 2PQ to find the percentage of the population that's heterozygous, okay? So the easiest way to do these, again, is to figure out what you're given and what you're looking for, and know that you need to use P plus Q equals 1 and figure out something that's going to make that usable for you, okay? All right, let's do some more practice problems so that we can see, because the biggest challenge with Hardy Weinberg is that what you're given and what you're looking for changes every time you do the problem. Okay, so whenever I talk to math teachers about how to teach Hardy Weinberg, blah, 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 they're like, oof, you just need to keep practicing with that, because there's a bunch of different ways that you can set this up. And the best way that you can do it is just to continue to practice.